Hello and welcome back to part 2 of our Unit 12, studying the DNA molecule. And in this section we are going to be looking at what actually comprises the DNA molecule. And before we can do that, we need to look at what does DNA actually stand for. What those letters stand for is deoxyribonucleic acid. That is the name of the actual molecule that you see on this screen at the far right. Over here you can see kind of the typical DNA molecule, nice twisted kind of a ladder, and then to the left of that you can see a part of it that is spread out, and you can see that the molecule is kind of complex, and that's what we're going to look at. But before we can do that, we need to talk about what is a nucleic acid. So acid itself is a type of molecule just like a fat or a carbohydrate or protein, and a nucleic acid is made up of little tiny pieces called nucleotides, and these nucleotides hook together into long chains, and then they wrap up, and that is what makes the nucleic acid itself. So now you should be asking yourself, what in the heck is a nucleotide? Well, these little tiny building blocks are made out of 3 parts. And you can see in the picture, a very simple picture. One is going to be a 5-carbon sugar. In this case for DNA, it's a deoxyribose sugar, and that's the blue. The next is a phosphate group, which we have stuck off here and symbolized by the orange circle. And the third is a nitrogenous base. And that's the part that we're going to talk about next. Before we do that, here's just another drawing showing the structure of a nucleotide. Over on the left, very similar to what we just saw, our 5-carbon sugar, and then you can compare that to the right. Sugar, being an organic compound, has carbon in it, and then just hydrogen and oxygen. You can see the green is the phosphate group with the phosphorus atom in the center, and then the blue at the top, the nitrogen base, has the nitrogen molecules. Now we have the most interesting part, at least in my opinion, of the DNA molecule, and it's the fact that the nitrogen bases come in only 4 types. So for as important as this molecule is to our very existence, it's actually pretty simple when we break it down. So the 4 bases are called adenine, guanine, cytosine, and thymine. And if you take a look at the diagram, you can see that the adenine and the guanine have the name purine above them. And the purine name is indicating that adenine and guanine actually have a double circular structure. The cytosine and the thymine are called pyrimidines, and they just have a singular ring structure. And you can think of these bases very similar to letters in the alphabet, where we have 26 letters in our alphabet and can come up with almost an infinite number of words. DNA has 4 bases to work with, and these 4 bases can be put together in a nearly infinite set of sequences and links to create every living organism that has existed or ever will exist. That's pretty impressive when you think that everything is only really different based on how these 4 bases are pairing up. Now the knowledge of these bases, the purines A and G and the pyrimidines T and C, was not a mystery. It was not unknown. And actually, Erwin Chargaff in 1950, 2 years before Hershey and Chase conducted their experiment, proposed these rules. And he had studied DNA and had analyzed these bases, and he found that no matter where he got the sample from, the percent of G and C were always balanced. And the same was true of A and T. There was always an equal amount of A and T in any sample. So, for example, if he found 20% adenine, he would find 20% thymine, and that meant 60% had to come from the other two bases. So 30 and 30 had to be the G and the C. Now, Chargaff had no idea why this was true, but because it always happened this way, it became known as Chargaff's rule. Using this information, I have a small assignment that I would like for you to do located on ebackpack. You have this diagram, and I just need you to examine this table, and based on what we talked about with Chargaff's rules, answer these 3 questions, and then have this submitted by 8 a.m. tomorrow. Make sure you get it submitted, and then we'll discuss it in class in case you need to make any changes. During the same time that Chargaff had made his rules, a young chemist by the name of Rosalind Franklin was doing some very interesting with DNA. And she was studying using X-ray diffraction. So what she would do, she would create a sample of DNA, and then she would twist these fibers and pull them out and lay them across her slides until they were nearly parallel. So she would stretch out these DNA, and then she would hit them with an X-ray, and then she would take a picture of it. And this X-ray, diffraction just means scattering, came out in this pattern that was always seemed to look like an X. And based on this, it appeared that there were 2 strands. A new word was coming from this. We were saying that these strands appeared to be twisted. So if we have this X pattern that repeats over and over again, if you can picture, it would have to twist. And in the biology world, anything that is twisted like that, we say it's helical or a helix. While Franklin was working on her X-ray diffraction work with DNA, 2 scientists were working. One was an American biologist, James Watson, and the other was a British physicist by the name of Francis Crick. And they were also trying to figure out the structure of DNA. And this was actually going on in 3 different parts of the world. Watson and Crick were working on it, Linus Pauling was working on it, and Rosalind Franklin was working on it, along with kind of her boss, Maurice Wilkins. And it was kind of a race. Who was going to be the first to come up with the correct structure of DNA? And Watson and Crick kind of had a laid-back approach to it. Yes, they wanted to be the first, but they didn't really do a lot of research. It was more a lot of tinkering. And their models were basically wire and cardboard, and they would use a ring stand and clamps. And they just could not get the structure to work. They knew the basic parts, that we had these bases and we had these phosphate groups, but they didn't know where to put them or what the structure looked like. And it was driving them out because they could not figure out where to put these bases. Put them on the outside, if you put them on the inside, how do you hold them together? And until Watson saw Franklin's X-ray picture and remembered that Crick had told him that a double helix model would look like an X, that he realized the correct way to put the molecule together. In their paper, Watson wrote, "The instant I saw the picture, my mouth fell open and my pulse began to race," because what they'd been working on for so long, he knew what he had to do. And to this day, Watson and Crick are the ones whose names come up when we talk about the double helix structure of DNA. When you discuss DNA, you always use the word double helix, 2 strands, helical meaning that they are twisted and they wind around each other. Here's another look at that same structure, just a little bit closer, that we have our sugar and phosphate on the outside, and we have our nucleotides pairing up in the middle, and you can see that there's bonding between them, holding them together. And this is often referred to as a twisted ladder, where the sides of the ladder are the sugar and phosphate, and they will alternate. So if you look at that picture, you can see the green and the orange stripes going up the green. And then the steps, or the rungs of the ladder, are your bases, always pairing up in the same way. You'll always have an A paired with a T and a C paired with a G, or vice versa, but you'll never have them pairing the other way. So the Watson-Crick model is the accepted model of today. It explained Rosalind Franklin's X-ray pattern. It explained Chargaff's rules of pairing. We say that the strands are anti-parallel, meaning they run in opposite directions. And we refer to the different ends of the DNA molecule as a 3' and a 5', and I will show you that on the next slide. It's also discovered that the bonds that held the bases together were very weak hydrogen bonds, and they had to be weak bonds because that DNA had to come apart when the cell would divide and the DNA would replicate. And the idea of A pairing with T and C pairing with G is now known as base pairs. So when we talk about base pairs, we're always talking about those letters coming together. Just another look at it here. Adenine will always be with thymine, so you remember at, and then guanine always pairs up with cytosine. I always think of it as go-kart, and those letters always go together. If you take a look over here on the right, you can see what we mean by the sides of the molecule being complementary. And that it means they are basically mirror images of each other. They're kind of an opposite. So if we have an A on one side, we'll have to have a T on the other. Where we have a phosphate group on one side, we're going to have a sugar molecule on the other. So each side of the DNA molecule is the opposite of the other side. All right, looking at this section right here, where we have one side of our molecule starting with the 3' and ending with the 5'. When we look at the other side, can you guess what would pair up here? So what would be across from A? What would be across from G? Did you guess correctly? A's will always be with T's, G's will be with C's, and so on. Well, in 1962, Watson and Crick were awarded the Nobel Prize in medicine for their discovery of the structure of DNA, and they actually had to share this award with Maurice Wilkins, who was working at the lab with Rosalind Franklin. So once again, we had a woman who was key in determining the structure of DNA who was left out of winning a Nobel Prize. And what we're going to do next is we're going to watch a movie that kind of talks about this whole discovery that went on with Watson and Crick and Wilkins and Franklin. And you'll see at the end why Franklin did not share in this honor, and then it's up to you to decide if it was fair or unfair.